Hi everyone, and welcome to the Forensic Evidence Recovery Process video lecture. Let's get started. In this lecture, we're going to discuss the process of evidence recovery at a crime scene. Now, there are four main steps in this process, starting with the detection efforts to locate evidence at the scene. We're then going to discuss how to collect, preserve, and submit various types of evidence so that it can be used in the courtroom. Detecting evidence is the first step of the forensic evidence recovery process, and I want to highlight a few suggestions here. Remember to focus on evidence that establishes relationships. The most valuable type of evidence establishes a threefold relationship of the victim and the suspect to the scene, and also to one another. But evidence is also valuable if it establishes a link between two of the three circles shown here. Of course, you do need to consider the established relationship between the suspect, the crime scene, and the victim, because that can affect what's relevant. Remember the example of a fingerprint on a knife that we've talked about several times. If a fingerprint on the knife is on a drawer located inside the suspect's home, it's not valuable to investigators because it doesn't establish a relationship between the suspect and either the crime scene or the victim. However, a fingerprint in blood on the knife in the suspect's home can establish a relationship between the suspect and the victim, and potentially the crime scene as well, depending on where the crime itself occurred. Another important consideration when searching for evidence is to consider Lecard's principle and to focus on potential points of contact. These areas are what's known as focal points because they have the highest likelihood of collecting relevant evidence. Focal areas include the entry to and the exit from the scene itself, likely suspect pathways through the scene, and any items touched or moved by the victim or suspect during the events. It's recommended to place a high priority on all focal points identified during the preliminary walkthrough of the scene. Now, that's not to say that you can't search for evidence or shouldn't search for evidence in other areas, but focal points are most likely to provide valuable trace evidence like footprints or fingerprints, things that can be easily damaged by investigators during the scene investigation. By focusing on these areas initially, we can hopefully reduce the amount of trace evidence that's lost during processing. Finally, remember to be aware of objects or materials that can be used as controls in later testing. If you're investigating an arson case, it's important to take samples from areas where it's unlikely that accelerant is used, so that the forensic chemist can compare control samples to the potential accelerant. This is one of the reasons why crime scene investigators must be knowledgeable about the laboratory analyses that forensic evidence undergoes, even if the investigator themselves does not have a laboratory position or does not analyze evidence in that manner. Only with a complete understanding of what tests can be run on what type of evidence can the investigator make sure that the evidence and any required controls are collected appropriately at the scene and submitted to the laboratory. Now, when detecting evidence, investigators should move through search techniques in such a way as to avoid damaging techniques until the end of the scene. In a previous lecture, you learned about how investigators should refrain from taking actions at a scene that alter or destroy part of it until they have completed a less intrusive search, like a visual search or searching with a flashlight. The same goes with evidence detection. Investigators should use non-destructive methods of detection first before they move on to methods that could potentially destroy or contaminate nearby evidence and scene features. Now, there are a variety of techniques that can be used to help investigators locate evidence at a crime scene. The most common and the least destructive is searching with light. Investigators can use a high-powered flashlight to help make evidence visible. For example, remember how useful oblique photography can be for highlighting impression evidence and fingerprints. Investigators can also detect a naturally luminescent material by viewing the crime scene in the dark. In most cases, though, the evidence itself is not going to luminesce on its own, but it might fluoresce in the presence of the right type of light. To fluoresce objects for detection, investigators use an alternate light source, or ALS for short. ALS uses a light source that's designed to emit a specific wavelength of light with high intensity. Investigators use filtered glasses to filter out the reflected wavelengths from the light source itself, allowing for the visualization of fluorescent emissions from the evidence. ALS emissions can be captured on cameras by using the same filter in front of the camera lens, just like you put the filter on your eyes. Now, something to keep in mind is that even though the ALS light source emits a very intense light, the resulting fluorescence is usually pretty weak, and it's best visualized and photographed in a dark room. The reduced intensity also creates problem with image capture of the fluorescence, because the camera is going to require a significantly longer exposure than normal in order to expose the fluorescent light well. The recommendation is to place your camera on a tripod and document the fluorescent with a series of photographs using longer and longer shutter speeds so that you can ensure you have a properly exposed photograph of the evidence for documentation. To create an examination quality photograph of a fluorescent image, you're going to need to use a fluorescent ruler as well so that the ruler is visible in the photograph as well as the evidence. Now, something to keep in mind about ALS is that most type of evidence will only respond to a certain wavelength. Unless the investigator is trying to focus on a specific type of evidence, attempts should be made to search the scene with all potentially useful light wavelengths. Some ALS kits will include bandpass filters corresponding to different wavelengths, so the investigator simply has to change out the filter in order to change the wavelength of the light emitted. Other ALS kits contain separate light sources for each wavelength. For example, the ALS you will have access to in this course is designed to emit light at 455 nanometers, making it useful for a general search by highlighting a diverse set of evidence type. So it will light up fingerprints, semen, gun shop residue, and fibers, but it won't be as helpful with evidence type like paint chips. If the investigators are purposefully targeting a specific type of evidence, they can use the corresponding ALS wavelength that's best suited for that particular type. The last type of light search I want to highlight is UV light searches. Ultraviolet light is a high-energy, short wavelength beam. UV light is categorized as either UVA or UVB. Now, UVB is hazardous to human health, so obviously we don't use that. Investigators rely instead on UVA light. Because UV light penetrates skin deeper than visible light, investigators have used long-wave UV light to detect bruises and bite marks, even when they're not detectable on the top layer of the skin. The research on the efficacy of this process, though, is mixed. Some research finds that UV light searches reveal bruises and injuries not visible with other light sources, with some published research claiming up to 5 months of detection after the bruising event. However, other research findings say that infrared photography and visible light photography are just as efficient or sometimes even stronger than UV detection, and there's really no clear consensus on this at this time. One additional benefit of UV light on human skin, though, is that UV light passes through tattoos and birthmarks, and so that can allow for better visualization of evidence like bruising that's located beneath a tattoo or a birthmark than visible light would. Of concern with UV light, though, is the fact that UV light will destroy or damage DNA recovery. So, as a result, this type of search should not be used unless absolutely necessary if you are trying to collect genetic material for analysis. Now let's switch our focus to the next step of the evidence recovery process, the collection stage. Like everything in crime scene, documentation is essential. Investigators should take extensive notes at the scene describing what they did, what they found, how they found it, and what they used to process and collect it. Taking photographs or video at each step of the scene processing can also help investigators document what happened and when. Investigators should also maintain a proper chain of custody from the time that they locate the evidence to the time that it's submitted to the laboratory or evidence locker. That way, the evidence can be vouched for as being safe from unlawful contamination or destruction. Deciding what evidence is important and what should be collected is a challenging process for investigators. On one hand, you want to avoid what we call the garbage truck approach, where you collect anything that could possibly be evidence. But at the same time, you only get one shot at crime scene processing. So if you skip over something and then it later turns out to be relevant, you've already missed your chance. For that reason, investigators should keep their minds open to all possible events that occurred at the scene. Even if the scene looks like an obvious suicide, take precautions and properly search and collect evidence as though it was an intentional homicide. That's going to make sure that you've secured the relevant evidence just in case down the road evidence analysis and reconstruction suggest that maybe it actually was a intentional homicide and it was just simply staged as a suicide. It's a way to cover your butt. Now, one of the resources that I used to create this video lecture is the Texas Crime Laboratory Division's manual, which was last updated in fall of 2024. They have a helpful quick reference for evidence collection and packaging table that gives you detailed recommendations for packaging based on the type of evidence and what specific examination requests that you would submit to the laboratory. I encourage you to use this packaging list as a cheat sheet during your packaging lab so you can reference as needed. Now, all types of evidence have a preferred way to collect them and a preferred type of container, but you're not going to be expected to memorize this full list of techniques and containers. But there is a short list provided in your readings so that you're just familiar with the different things that crime scene investigators consider during evidence collection. First and foremost, though, is this consideration. If you are able to, it is preferred that you collect the entire item itself and save all the processing until the item is submitted to the laboratory for analysis. Collection of the entire substrate gives analysts the most material to work with, and that can be really important if they're later going to use destructive methods of analysis like gas chromatography. Delayed processing until the item has been removed to the laboratory can also help with trace evidence collection, because there are lots of field processing techniques that impede the use of future analyses. By taking the item to the laboratory, an analyst can conduct a series of analyses in the proper order to make sure that each analysis does not limit future analysis options. For example, the flowchart here on this slide is from the appendix of the Complete Crime Scene Investigation Handbook, this huge text that goes really deep into each specific type of evidence in its packaging and its analysis. And they have a flowchart in there that documents the list of analysis used for latent fingerprints on plastic surfaces. Now, the chemical reagents here are listed in the order that they can be analyzed in. So, if the analyst starts with the first recommended analysis, which is Sudan black, then they can work their way down to alternative chemical analyses like cyanoacrylate or small particle reagent. However, if the analyst chooses to skipping all the top, going straight down to small particle reagent, they can't go back up the chain and test the material using things like Sudan black. So that's going to limit the potential tests that they can use to capture important fingerprint evidence. There are a myriad other collection methods, including tweezing, tape lifting, cutting, pipetting, and more. The type of collection method is going to depend on the type of evidence that you're trying to collect. Tweezing is very useful for trace evidence that's visible to investigators. Tweezing gives the investigators very specific evidence to analyze, and it doesn't give us a lot of extra data. However, forensic vacuuming, also helpful for trace evidence, but forensic vacuuming collects everything in a particular location, so it creates a lot of extra data that ends up maximizing the amount of time that evidence processing takes. However, it's a very effective method of capturing trace evidence, even trace that's too microscopic for investigators to see with the naked eye at the crime scene. So you have to decide what's best in your specific case under your specific circumstances. Pipetting, casting, and scraping also have more specialized purposes. For example, they're best suited for liquid, impression, and paint evidence, respectively. Like the light searching techniques, I don't expect you to memorize what type of collection method works with what type of evidence, but I want you to understand the different types of collection methods available so that you can then make appropriate judgments about what you're going to do to collect evidence when you're faced with the scene description. One of the previous textbooks I used for this course had a really good step-by-step method for evidence packaging that I want to share with you. So first, they recommended that they leave evidence containers at the crime scene entry point. As you identify evidence inside the scene, package the evidence on the spot in the appropriate container. Close the container and label the package. Then, take the evidence outside of the scene boundary, seal it, and store it at a designated location. If you have multiple investigators at the scene, one or more investigators can take on the role of evidence officer. These individuals will be in charge of maintaining the chain of custody for all of the evidence and for sealing all of the evidence packages. The final topic I want to discuss in this lecture video is the process of submitting evidence to a laboratory. When submitting evidence to a crime lab, the investigator will fill out a request for lab examination form, or something similar depending on the specific agency's protocols. On the request form, the analyst will describe the evidence that's submitted and explain what type of tests and analyses are requested. Now, that doesn't mean that only those tests will be conducted, but this request form will help crime laboratories determine which analyst the evidence should be submitted to first and in what order the evidence should be processed. Shown here is the laboratory submission for the Texas Department of Public Safety's Crime Lab. You can see on page 2 there's a table where you can put the evidence description of what you're submitting, and then you can add specific request codes identifying the type of forensic analysis that you are requesting for each piece of evidence. When submitting evidence, it's important to remember that you should not submit just to submit. Forensics laboratories do have limited storage space. So if you bring a whole moving truck full of evidence and it's a burglary, your lab's not going to have room to store the evidence from an actual homicide. So remember that forensic laboratories are also typically under severe budgetary strain. So you have to be selective on what evidence you submit for analysis. Focus on the evidence that is relevant to the case and try to assess which pieces of evidence are most likely to produce useful results. You must also consider the type of case and the case priority when selecting the evidence to analyze. Each agency could potentially have rules that limits the number of evidence items that can be submitted and the number of specific analyses that can be requested based on the type of case. Now, to give you an idea of the limitations, here are some of the limitations that are specific to the Texas Physical Evidence Collection Handbook. This handbook breaks down each type of analyses and provides specific evidence restrictions. For example, here are some of the limitations for DNA evidence. For burglary and property crimes, you're limited to 2 items only for DNA testing. Other crimes against persons that are not burglary or property crimes are allowed up to 5 items, and homicides are limited to 10 items. However, the 10 items for homicide must be prioritized because the original examination will only be completed on the top 5 priority items. If DNA is identified on any of these 5, the remaining pieces of evidence will not be examined. But if the priority 5 homicide items do not yield usable DNA, then the analyst will move to the remaining 5 items. Sexual assaults have a similar caveat. The original test is restricted to 3 specific items: the sexual assault evidence collection kit, 1 pair of underwear, and 1 condom. If all 3 of these items do not yield usable DNA results, then investigators can submit up to 5 additional items for testing. If one of the 3 items contains a DNA sample that can be tested, then no additional evidence can be submitted. Now, in all cases, there's some flexibility. You can always submit additional items for DNA analysis, but the submission requires written justification, and it may require approval from case prosecutors. The idea is that the state wants to reserve DNA testing for those items that are most likely to produce a DNA fingerprint and that are also essential for the prosecution of the criminal case, which is why you're asked to get approval from the case prosecutors. Another thing to keep in mind when submitting evidence to a laboratory is that the evidence must be submitted to the right laboratory. This image is from the 2024 updated Texas Physical Evidence Handbook as well. Each column represents a different crime laboratory in the state of Texas, and the X's mean that the laboratory contains the equipment and the analysts for that particular type of evidence analysis. Excuse me. So, for example, if you have a drug sample that requires a toxicological analysis, the only lab in the state that does that is in Austin, which is the main crime lab for the whole state. If you collect bullet cartridges for APHIS, you have to send that to Austin too. If you want a DNA sample analyzed, though, you can use multiple laboratories, including Austin, you can go to Houston, El Paso, or Waco. In Tyler, we have the capabilities for seized drug analysis, alcohol toxicology, and some firearms and tool marks analysis. But we don't have any trace evidence capabilities. So it's important that the investigator know what laboratory services they have access to and where those services are located. And that is all going to be based on agency location and submission protocols. All right, well, that completes this video lecture. Thanks for watching, everyone.